Hello buddies, welcome back to the channel. Have you ever wondered how a tiny single cell becomes a complete human body with trillions of cells? How does one cell know when to grow, when to copy its DNA and when to divide? The answer lies in the one of the most fascinating process in biology, that is cell cycle and then cell division. The chapter which one we are going to revise today, that is the day 7. And remember, questions from this chapter are very common in NEET examination. So today we will understand it in very simplest and then smartest way. So let's get started. And here comes, before that I just want to revise you the stages of the cell cycle. See, cell cycle has the interphase and then M phase. It is totally divided into, cell cycle is totally divided into interphase and then M phase. Okay, now once interphase is having 3 phases: G1 phase, S phase and then G2 phase, gap 1, gap 2 and then synthesis phase. Okay, G1 phase is said to be a growth phase, whereas S phase is said to be a replication or a synthesize phase where the DNA is going to be synthesized, and then G2 phase is said to be what? Growth phase. And here again the M phase where the cell division is going to take place, and then M phase, that is mitosis or else meiosis. Mitosis is having again the 4 stages, again the 4 steps, that is called as prophase, metaphase and then anaphase. Here the small trick to keep in mind, that is PMAT. P refers to the prophase, M refers to the metaphase and then A refers to the anaphase and T refers to the telophase. Okay, for these also I will give you tricks in upcoming videos, so watch the video till the end. Now let's get started with the first question, that is question number 1. All cells reproduce by dividing into dash, with each parental cells giving rise to dash cells each time they divide. How many? One cell reproduce by dividing into how many, and each parent cells giving rise to how many cells? Okay, option A 1 and then it gives 4 daughter cells, option B 2 and then 2 daughter cells, option C 1 and then 2 daughter cells, option D 2 and then 4 daughter cells. Here the right option is B, 2 and then 2 daughter cell. One cell reproduced by dividing into 2 with each parental cells giving rise to 2 cells each time they divide. So here the right option is option B. Next question from the cell cycle. Second question. A cell cycle comprises all the listed events except A. Cell growth, B. DNA replication, C. Transcription and then D. Cell division. And here the right option is option C, transcription. Transcription is not a listed event of the cell cycle. Question number 3. Consider the following statements. Statement 1, that is cell growth in terms of cytoplasmic increases is a contingent process which occurs during cell cycle. Statement 2, DNA synthesis occur only during one specific stage in the cell cycle. Statement 3, the event of cell cycle are under genetic control. So here the option are given A, B, C and then D. If the option statement 1 is false and then statement 2 and then 3 are true. Statement 1 is false and then remaining 2 are true. Phases of cell cycle. Fourth question. Cell of human divide once in approximately, human cell divides approximately, option A 60 minutes, B 90 minutes, C 24 hours, D none of this. So here the option is C 24 hours. Human cell divide 24 hours, means one cell it takes 24 hours to divide, whereas if I say example for the bacteria E. coli, it takes only just 20 minutes to divide. Fifth question, read the following statements and choose the correct option. Statement A, the M phase represents the phase when actual cell division occurs. Statement B, interphase represents the phase between two successive M phases. Yes, they both are correct. Both these statements are correct here. So the option D is right. Next question, that is question number 6. An average duration of yeast cell cycle is option A 60 minutes, option B 90 minutes, option C 20 minutes, and then D 1 day. So here yeast cell cycle takes 90 minutes, that is yeast Saccharomyces cerevisiae we used to call, and a fungi which takes 90 minutes to divide. One yeast will give you 2 yeast after 90 minutes. Seventh one, cell cycle is divided into how many basic phases? Cell cycle is divided into how many basic phases? 1, option B 2, option C 3, 4, option D 6. So here the right option is 2, interphase and then M phase. Eighth one, which of the following is or are enlisted and basic phase of cell cycle? Option A G0 phase, B S phase, and then C interphase, and then D metaphase, and then here the right option is option C interphase. Ninth question, the phase of cell cycle during which mitosis occurs is option A interphase, option B M phase, option C G phase, option D S phase, and then here the right option is M phase. M in the sense it is mitosis if it is the mitotic stage. 10th question, the phase between two successive M phase. Option A interphase, option B G phase, option C S phase, option D M phase. So here the right option is interphase. 11th one, the time span of interphase and then M phase is an average human cell cycle is option A 12 hours each, B 19% M phase and then 1 hour interphase, C 8 hours M phase and then 16 hour interphase, and then D 1 hour M phase and then 23 hours of interphase. So here the right option is D M phase. Mitotic phase takes only 1 hour, whereas 23 hours interphase will take, whereas cell is grow and then it is going to be synthesized, again somewhat it is grows, and then next it is entered to the mitotic phase for the prophase, anaphase, metaphase, anaphase, and then telophase. Second, 12th one, the correct sequence of cell is option A M G2 S and G1, B S G2 S1 and M, C M G1 G2 and S, D G1 S G2 and M. So here the right option is option D, G1 first, next G S, then next S G2 and then M. Gap 1 synthesis phase, and then gap 2, and then next comes the M phase. To remember this, grow, synthesize and grow again. Then next go for the mitosis. Can you remember that? Yes. Interphase has 3 stages, G1, S and then G2. Trick to remember is grow, synthesize and grow again. After that you may go for the mitosis. Got it? G1 grow, S synthesize, and G2 again it is going to grow. DNA synthesis takes place in the S stage. 13th question, match the following column 1 with column 2. G1 phase, metabolically active cell do not proliferate. S phase, G0 phase and then G2 phase. So here the right option, if you know any of the two, if you know any of the two, you can get the answer. For example, if I know S phase, okay, S phase, content of DNA is going to be doubled. So B is 2. If I go B is 2, this option A and then B has B2. So C is cancelled here. C is wrong, obviously. Next, we'll go with G0 phase. G0 phase is metabolically active, do not proliferate. G0 phase will be in a silent mode only, but it will be metabolically active. So C is 1. So here the right option will be A. Like that. In a match the following, if you are confident with any of the two options, by the eligibility method you can get the answer very easily. Don't waste time there. 14th question. The process which mark as a start and usually end of M phase are option A, division of cytoplasm and karyokinesis respectively. B, cytokinesis and division of cytoplasm respectively. C, separation of daughter chromosome and cytokinesis respectively. D, karyokinesis and karyokinesis respectively. The process which mark as a start and usually end of the M phase are separation of daughter chromosomes is a starting point, and then cytokinesis means the division of the cytoplasm is the end, that is the start and then end part of the cell cycle. 15th one, resting phase of cell cycle is A, M phase, B, interkinesis, C, G1 and G2 phase, D, interphase. So here the right option is interphase. Next question, 16th one, interphase is divided into dash phases further. Interphase is divided into dash phases further. Option A 4, option B 3, C 2, D is 5. Here the right option is option B 3. G1, S and G2 phase. Grow, synthesize and grow again. 17th one, which of the following stage corresponds to the interval between the mitosis and initiation of the DNA replication? Which of the following stage correspond to the interval between the mitosis and initiation of the DNA replication? That is G1 phase. M phase, between the mitosis and then initiation of DNA replication comes what? G1 phase. G1, yes, G2, M phase. No, after M phase again G1 phase will come, isn't it? So in between the S and then M, which one will be there? G1 phase. So the correct option is D. Next, 18th one, select the correct statement about the G1 phase. A, cell is metabolically inactive. B, DNA doesn't replicate. C, DNA replicate. No. D, chromosome number is doubled. No. Here the correct statement is B, DNA does not replicate here. Here the cell in G1 phase, cell will be metabolically active. DNA doesn't replicate. It is the correct statement. 19th one, correct sequence of phase of M phase is, M phase stage, that is mitotic phase stage is A, cytokinesis, prophase, metaphase, anaphase, telophase. The first option itself is right. How you can remember this one? Let me give you a trick. PMAT, in simple words you can remember in the form of PMAT. First, in the first slide only I said PMAT, P for prophase, M for metaphase, A for anaphase, and then T for telophase. Or else you can also keep in mind a statement that is please make a team. Please make a team. Here please refers to prophase and M will for metaphase, A for anaphase, T for telophase. Got it? Either you may keep PMAT as a shortcut, and then either you may keep a please make a team as a shortcut trick. Got it? Yes. Next, 20. What would be amount of DNA? Amount of DNA is indicated in the form of C, and the number of chromosome N in animal cell just after the completion of S phase. If the initial amount of DNA is 2C and then number is 2N. Keep in mind, amount after the S phase, completion of the S phase, amount will double, okay, but the number won't. Number will remain same. If one chromosome is there, then the chromosome will be only one only. If 2N is given, only 2N will be there, but the amount will be doubled. If 2C is there, means it will become 4C. So what will be the right option? 4C is here also and here also, but the N is given 4, so B option is wrong and the right option is C. Got it? Amount will be doubled, number will remain same. Keep in mind, after the completion of S phase, after the duplication of DNA, the amount of DNA will double, but the number of chromosome remains same. That's it. 21. Duplication of centriole occur in duplication of centriole. Centriole is the presence of it's a unique characteristic of animal cell where they are going to release the asters in animal cell. But in plant cell, the formation of spindle fibers will take place a specific area which is called as asters. Okay, so here duplication of centriole occur in M phase, B G2 phase, C S phase, D G0 phase. So the right option is C S phase. Duplication of centriole and then duplication of amount of DNA is both occurs in the S phase only. Next, 22. The S phase of animal cell marked by option A DNA replication. You can't mark in animal cell. Why? Because DNA replication also takes place in plant cell. B centriole duplication, exactly. And then C cell growth and protein synthesis, and then D A and B. So here the right option will be option D, A and B. DNA replication and then centriole duplication. Of course, DNA replication takes place in plant cell also and animal cell also, but the centriole duplication will be only seen in animal cell. So that is marked by what? Centriole duplication as well as the DNA replication. 23 question. The G2 phase of cell cycle is pronounced by A cell growth and division, B cell duplication, C protein synthesis and centriole duplication, D cell growth and protein synthesis. So it is pronounced by cell growth and then protein synthesis. The proteins which are helpful for the M phase, those all are going to be synthesized in the G2. 24. Find the mismatched column 1 with column 2. A karyokinesis, separation of daughter chromosomes, right. B cytokinesis, division of cytoplasm, correct. C interphase, smallest phase of the cell cycle. No, smallest phase is the M phase, which takes only 1 hour, but the remaining 23 hours is of interphase only. G1, yes, and then G2. So here the right option is C. 25. The inactive stage of cell cycle is option A quiescent stage, option B G1 phase, option C S phase, option D A and B. Here the inactive stage is option A quiescent stage, or else you also called as that one as the inactive stage of the cell, quiescent stage. 26th one. Choose the correct statement with respect to G0 phase. A also known as quiescent stage and start after G2 phase. B cell of this stage remain inactive and no longer proliferation. C cells of this stage remain inactive but no longer proliferation unless called to do so depending on the requirement of the organism. D cell of this stage remain active and proliferation till death without any condition. So here the correct statement with respect to G0 phase is option C. 27. How many chromosomes does onion's somatic cell have? Chromosome number of onion is 16. 28. What number of chromosome does onion's somatic cell have in G, S, G2 and M phase respectively? G, it is having 16, whereas S number won't change, amount will change, so it is also 16, and then G2 16 and M phase is 16 respectively. 29. Mitotic division occurs in A. Diploid somatic cell, B. Haploid male honeybee, C. A and B, D. Gametes. So here the right option, mitosis occurs in diploid somatic cells. Mitosis is for the somatic cells, and then whereas the haploid cells and then for the gamete formation, meiosis will take place, where the number of chromosome is going to be halved. Next question, question number 30, M phase. 30th question, M phase refers to A. Metaphase, B. Meiosis, C. Karyokinesis, D. A and B both. So here the right option is karyokinesis. 31. Correct order of mitotic division is, correct order of mitotic division is, I said PMAT. P, whether first P is there? No, first option A is wrong then. P match. So right option is B. So how simple it is. 32. Most dramatic period of cell cycle is, most dramatic period of cell cycle is A. Gap 1 only, B. M phase, C. S phase and then D. interphase. So here the right option is B. M phase. It is the dramatic period of the cell cycle because it changes very fastly. The cell is going to be change its physical condition. 33. Equational division refers to A. Meiosis, B. Mitosis, C. Number of cell chromosome in parent and progeny cell is same, and then D. B and C both. So here the right option is B. B and C both are refers to the equational division. Mitosis is a equational division, meiosis is a reductional division because the number of chromosome is going to be reduced in the meiosis. 34. Karyokinesis of mitosis is divided into 4 stages. Dash stages. Option A 2, option B 3, option C 4 and then option D is 8. So option C 4 stages. Already I said about that. Sorry for that. 35. From prophase. Now starts the questions from stage wise. So first stage, that is prophase. 35. Select the correct option. 1. Prophase is first stage of karyokinesis. 2. It occurs after completion of protein synthesis during the cell cycle. So here the right option is A because both the statements are correct. 36. Asters formed during prophase are A. Composed of microtubules, originate from centromere. Option B. Composed of protein which secreted by the Golgi body. C. Highly condensed area of chromosome, and then D. None of this. So here the right option is composed of microtubules, originate from centromere. 37. During the prophase, which of the following occurs? A. Condensation of chromosome material. B. Chromosomal material becomes tangled. C. Chromosome duplication. D. Movement of both centriole at one pole to one pole of cell. So here the right option is option A. Condensation occurs in prophase. Condensation of chromosomes means whatever the chromatin network will be there, no, that chromatin network is going to be condensed in the prophase. And then chromosomes formed and then aligned in the equator plane in metaphase, and then daughter chromosomes are going to be separated in the anaphase, whereas again the decondensation of the chromosome takes place in telophase. So here 38th one, choose the incorrect match. A, beginning of movement of chromosome to opposite poles. Opposite poles is for anaphase, that is not for the prophase. B, two asters with spindle, that is mitotic apparatus. C, attachment of spindle, metaphase. D, chromosome move to opposite poles, metaphase. So here the right option is A, beginning of movement of chromosomes to the opposite pole. It is for the anaphase, not for the prophase. 39th one, mitotic apparatus consists of A, 4 asters with spindle fibers. B, 1 aster with spindle fibers. C, 2 asters with spindle fibers. D, centrosome with their microtubule without spindle fibers. Mitotic apparatus consists of 2 asters with spindle fibers. Always there will be 2 asters. 40. How many of the following structures are observed when cells are viewed under the microscope at the end of prophase? Golgi body, ER, nucleolus, nuclear envelope, and then centrosome. Here the right option is 0, because all of them are going to be disappear by the end of prophase. Only the condensed form of chromosomes is going to be appear in the end of the chromosome. 41. How do the chromosome appear during prophase of animal cell during mitosis? A, consisting of 4 chromatid which remain attached to centromere. B, consisting of 2 chromatid which remain attached to centromere. C, consisting of 4 chromatid without centromere. D, as chromatin material without any defined structure. So here the right option is option D. Next, what difference would indicate early prophase and late prophase of animal cell? Early prophase and late prophase, what are the differences? A, nucleolus and nuclear membrane present. Nucleolus and nuclear membrane are absent. B, chromosome are highly condensed. There is no condensation of chromosome. C, nucleolus and centrosome are present. Nucleolus and centromere are absent. D, other organelles like ER Golgi body complex are not observed. Other organelles like ER Golgi complex are observed. So here the right option is option B. Chromosomes are highly condensed and then there is no condensation of chromosomes. That 43. Identify correct stage of given diagram. This is the one diagram and then this one is one more diagram. This is the nuclear membrane which is going to be disappearing and then there is somewhat chromosome is going to be condensed, and these are the centrosomes. A, late prophase and then early prophase. B, early prophase and then late prophase. C, metaphase prophase, and then D, prophase and metaphase. Here the right option is B, early prophase and late prophase. This is the late prophase and then this one is early one. Now the questions from metaphase. 44. The complete disintegration of nuclear envelope mark starts of A, late prophase, B, metaphase, C, anaphase, D, none of this. So here the option is B, metaphase. 45. Identify the stage. This stage is given, identify that one. A, transition to metaphase, B, anaphase, C, metaphase, and then D, telophase. Here the right option is C, metaphase. They are going to align at the metacentric plate, so it is of the metaphase. This is also metaphase. Identify stage of given diagram. Diagram 46th question. A, early prophase and then metaphase. B, late prophase, transition to the metaphase. C, early prophase and transition to metaphase. D, late prophase and metaphase. Here the right option is D. This is the late prophase and this one is metaphase. 47th question. Which stage of cell cycle is best to study chromosome morphology? A. Late prophase, B. Early prophase, C. Anaphase, and then D. Metaphase. So here the right option is metaphase. Metaphase is the best study to chromosome morphology because there will be aligning in the metacentric plate. 48th, condensation of chromosome is completed in A. Stage where chromosome is duplicated, B. Stage where DNA content doubled, C. Stage where complete integration of nuclear envelope occurs, D. Stage where complete disintegration of nucleus envelope occurs. So here the right option is D. 49. Metaphase chromosome is made up of A. Two non-sister chromatids which are held together by a centromere, B. Four sister chromatids which are held together by a centromere, C. Two sister chromatids which are held together by a centromere, and then D. Four non-sister chromatids which are held together by a centromere. Here the right option is C. Two sister chromatids which are held together by centromere. Let me show you what is. See, if I zoom this one, let me zoom. In this picture it will be very fine. This is one sister chromatid and this one is one more sister. They are attached to the centromere. Okay. Two sister chromatids which are held together by centromere. This is one sister chromatid and this one is one more sister chromatid, and they are held together with a centromere. 50th one. Kinetochore are A. Precursor of microtubules, B. Sites of attachment of spindle fiber, C. Site for origination of spindle fiber, D. Small disc shaped structure at the telomere of chromosome. So here the right option is option B, site of attachment of spindle fiber. See, for the chromosome what happens, if I saying this is the chromosome, one chromosome, there will be a small area here in the centromere, and that is said to be what? Kinetochore, and where the kinetochore is helpful for the attachment of what? Spindle fiber. This is the area where it is said to be what? Kinetochore, and this is very important for you. Next one. Metaphase is characterized by A. Some chromosome coming to lie at the pole. No, metaphase, the chromosomes are not at pole, they will be at the equator. B. One chromatid of each chromosome connect by its centromere to the spindle fiber from one pole. No, not only from one pole, they will be attached with both the pole. C. Sister chromatid connect by its kinetochore to spindle fibers from the opposite poles. Yeah, this one is the correct. So correct answer is C. From the anaphase stage, the questions are from here. 52. At the onset of anaphase, each chromosome split into A. One chromatid, B. Four daughter chromatids, C. Two daughter chromatids, and then D. 8 chromatids. So here the answer is two daughter chromatids. How come two daughter chromatids? If I say this is a cell where anaphase is going to take place, and then metaphase at the equator, chromosomes is going to be aligned. From the pole what happens, spindle fibers are going to form, and then they are going to attach to the kinetochore. Okay, and there will be a sister chromatid, and then they are going to pull towards the pole. This is from one chromosome and then this is from another chromosome, like this. So it is going to be pulled to this pole, and then this is going to be pulled to that. So there will be formation of two daughter chromosomes from one chromosome. Next question, 53. Anaphase is characterized by 1. Migration of daughter chromatid towards equator. No. 2. Centromere of each chromosome remain directed towards the pole. Yes. 3. Centromere of each chromosome remain directed towards equator. No. 4th. Chromatid split and centromere separate. Yes. 5th one, chromatid separate after the centromere split. So here the right one is 2 and then 5th one. 54. Identify the stage. This is the, just now I said, A, anaphase. Yes, exactly. The given figure is anaphase. Telophase. 55. During telophase, A, statement 1, chromosome clusters at opposite spindle poles. 2. Two daughter nuclei formed. 3. Chromosomes lose their individuality. 4th, it is a reversal of prophase. And then 5, nucleolus is not reformed. Choose the incorrect statement. Here the incorrect statement is 3rd and then 4th one. 56. Match the following column 1 with column 2. Syncytium. Divide the cytoplasm of animal cell. Cell plate occur in liquid endosperm of coconut. Cell furrow. Method of cytokinesis. Here the right option is B. A is 2. Syncytium means occurrence in liquid endosperm of the coconut. Whatever the endosperm of coconut we drink, no, that is not but the syncytium form of the nuclear division. There will be a rapid nuclear division in them. Next, 57th question. Cell plate represents A, primary lamella, B, middle lamella, C, both, D, formation of plate by lysosome. Here the right option is middle lamella. Next, cytokinesis. 58th one. Mitosis usually results in option A, haploid daughter cells with identical genetic complement. B, growth of multicellular organism. C, diploid daughter cells without identical genetic complement. D, haploid daughter cells without identical genetic complement. So here the right option is A, haploid daughter cells with identical genetic complement. 59. Which one is odd with respect to significance of meiosis? Option A, increase genetic variability in organisms. B, helps in restoring of original chromosome number in a sexual reproducing species. C, ensures production of haploid phase. And then D, cell repair. So here the right option is with respect to significance of meiosis is D, cell repair. 60th question. The growth with plant is or are contributed by A, mitotic division in apical meristem, B, meiotic division in lateral meristem, C, meiotic division in apical meristem, D, A and B both. So here the right option is A, meiotic division in apical meristem due to which the plant grows continuously. 61 question. A nucleocytoplasmic ratio in organism is restored by mitosis. B, the cells of the upper layer of epidermis, cells of lining of gut and then blood cells are begin constantly replaced by mitotic division. Choose the correct option from the above. Here the correct option is C, where the statement A and B both are true. From now onwards, it starts with a meiosis. 62. Meiosis results in A, production of gametes, B, reduction in number of chromosomes, yes, C, introduction of variation. This is also correct. So the right option is option D, all of this. 63. Meiosis ensure the production of dash phase in life cycle of sexual reproduction organisms, whereas fertilization restores the dash phase. A. Haploid and haploid. B. Haploid and diploid. C. Diploid and diploid. D. Diploid and haploid. So here the option D, diploid phase of life cycle of sexual reproducing organism where fertilization restored by haploid phase. 64. Which of the following statement is correct? A. Meiosis involves single cycle of nuclear and cell division. B. Doubling of chromosome occurs once during S phase. C. Recombination between sister chromatids of non-homologous chromosome. D. Pairing of homologous chromosome. Among them, D is the correct one. 65. At the end of meiosis 2, how many haploid cells are formed? A. 1, B. 2, C. 0, D. 4. So, right option is D. 4. From one cell, by the end of meiosis 2, we will be getting 4 haploid cells. Next question, that is question number 66. Recombination occurs between A. Sister chromatid of non-homologous chromosome. No. B. Non-sister chromatid of non-homologous chromosome. No. C. Sister chromatids of homologous chromosome. No. D. Non-sister chromatids of homologous chromosome. This one is right. The correct option is D. Typing mistake, make it D. See here, what is non-sister and then sister? Let me explain. See, this is the gray color, whatever the light gray color is there. This is one homologous chromosome and this one is another one. Okay, 2 homologous chromosomes are given. And this is one sister chromatid, this one is one more sister chromatid of this homologous chromosome, and this one is the one chromatid and this one is another chromatid of the another chromosome. When they are going to be crossing over, that's what it is called, what? Non-sister chromatid. See how come the gray, light gray is having the dark one and then dark gray is having the light one. The characters are exchanged. So that's what non-sister chromatid of homologous chromosomes are recombined. So this is the not the option C, option D is right. Please make a correction here. Option D is right. 67th question. The longest phase of meiosis is A. Prophase 1, B. Prophase 2, C. Metaphase 1 and then D. Telophase 2. So here the right option is prophase 1 because it is having again the 5 substages. That's what the prophase 1 is the longest phase of meiosis. 68. During which of the given phase homologous chromosomes separate while sister chromatids remain associated at their centromere? A. Anaphase of mitosis, B. Anaphase 2, C. Anaphase 1, D. Metaphase 1. So here the right option is anaphase 1. 69. Prophase 1 of meiosis is divided into dash phase based on chromosomal behavior. Just now I said they are divided into 5 phases. That is leptotin, zygotin, pachytin, diplotin and diakinesis. Here the trick to remember. Lazy zebra plays deep drums. Lazy zebra plays deep drums, which is you can remember for the leptotin, zygotin, pachytin and diplotin and then diakinesis. Now again I am giving you a short method to remember each stage main thing. in leptotin what happens? Chromosomes become visible. Okay, in leptotin chromosomes becomes visible. In zygotin synapsis begins. Synapsis begins. In Pachitin crossing over occurs. This area is mostly asked question where crossing over is going to occur. Okay na, Pachitin. Okay, in diplotin chiasmata visible. In diplotin chiasmata visibles, and in diakinesis terminalization is going to be occurred. So this is the most important thing which I can give you. So keep in mind the trick is lazy zebra plays deep drums. Lazy means leptotin, zebra means zygotin, plays Pachitin, deep diplotin, and drums diakinesis. Here chromosomes becomes visible, synapsis begins, crossing over happens, chiasmata visibles, and then terminalization. 70th one, identify correct sequence of prophase. Just now I said A. Leptotin, diplotin, zygotin. First lazy L it must be, isn't it? So the right option is none of them, because there is only given 3 3 3. Actually prophase has how many? 5 stages. 71, dyads of cells are formed in A. Telophase 1, B. Telophase 2, C. Diakinesis, D. Both A and B. So here the right option is telophase 1. 72, a bivalent is option A. Pair of non-homologous chromosomes, B. The complex formed by a pair of synapsed homologous chromosome, C. Formed during Pachitin stage, D. More clearly visible at zygote stage. Bivalent is the complex formed by a pair of synapsed homologous chromosomes. 73, synaptonymal complex dissolves during A. Leptotin, B. Diakinesis, C. Zygotin, D. Diplotin. So here the right option is diplotin. 74, during which phase of meiosis centromere splits? A. Anaphase 1, B. Anaphase 2, C. Telophase 2, D. Telophase 1. So here the right option is anaphase 2. 75, choose the correct option with respect to leptotin. A. It is the foremost and then short-lived stage of prophase. 2. It begins when the process of compaction of chromosome is accomplished. 3. Chromosome become visible under light microscope. 4. It followed by zygotin. So here the right option is C. 3 and 4 are correct while 1 and 2 are incorrect. 76. Zygotin is characterized by 1. Chromosome starts pairing. 2. Non-homologous chromosomes pair. 3. Synapsis occurs between the homologous chromosome. 4th, in formation of synaptimal complex in the homologous chromosome. 5. Formation of synaptimal complex in non-homologous chromosome. So here the right option is 1. 1, 2 and 5 are characterized by the zygotin. 77. Bivalent stage is A. Complex formed by a pair of synapsed homologous chromosome. B. Complex formed by a pair of synapsed non-homologous chromosome. And then C. Complex formed by 4 pair of synapsed homologous chromosome. And then D. Complex formed by 4 pairs of synapsed non-homologous chromosome. And then right option is option A. Complex formed by a pair of synapsed homologous chromosome, which is called as bivalent stage. 78. Crossing over occurs in. I said about this. Option A. Leptotin, B. Zygotin, C. Pachitin, D. Diplotin. So Pachitin where the crossing over occurs. 79. Pachitin is a stage that is or are A. long-lived than zygotin, B. 2 chromatids of each bivalent chromosome becomes distinct, C. short-lived than leptotin, D. Long-lived than leptotin and short-lived than the zygotin. Here the right option is A. Long-lived than zygotin. 80th question. Choose the correct statement from the following. A. Pachitin is characterized by appearance of recombination nodule. B. Recombination nodule is site of crossing over. C. Both A and B. D. Recombination nodule formed in diplotin. So here the correct statement is C. Both A and B are correct. 81. Given below are statement 1 to 6. Choose the correct set with respect to crossing over. Statement 1. It occurs between sister chromatids of homologous chromosome. 2. It is enzyme mediated process. 3. Recombination enzyme involves in it. 4. It occurs at a recombination nodule. 5. It occurs between non-sister chromatid of non-homologous chromosome. And then sixth one, it occurs between sister chromatid of non-homologous chromosome. Here the correct set of crossing over is 2, 3 and then 4. 82. Diplotin is not characterized by option A. Dissolution of synaptonymal complex. Option B. Tendency of recombined homologous chromosome of tetrad to separate from each other except at sites of crossover. C. Formation of chiasmata. D. Tendency of recombined non-homologous chromosome of bivalent to separate from each other except at sites of crossover. So here the right option is D, which is not characterized by the diplotin. Next question, 83. Question number 83. Chiasmata is A. X-shaped structure, correct. B. Formed by recombined chromosome yet to be separated, yes. C. Site of crossover, correct. So the right option is all of this. 84. Which stage of meiosis 1 lasts for months or years in some vertebrate oocytes? Option A. Diakinesis, B. Diplotin, C. Pachitin and then D. Zygotin. Here the right option is option B. Diplotin. 85. Diakinesis marked by A. Terminalization of chiasmata, B. Chromosomes are fully condensed, C. Meiotic spindle assembled, D. All of this. So here all of these are correct. 86. Meiotic spindle assembled to prepare A. Non-homologous chromosome separation, B. Formation of aster ray, C. Homologous chromosome separation, D. Both B and C. Here the right option is C. Homologous chromosome separation. 87. Match the following. The stages are given and then their formation. Leptotin, compaction of chromosome, zygotin, separation of chromosome, pachitin, terminalization of chiasmata, diakinesis, appearance of recombination of nodules, and diplotin, synapses. I said about these all stages so that you can combine them. So here the A option is correct, where one is A, zygotin is for synapses, and then pachitin is for appearance of recombination of nodules, diakinesis is for terminalization of chiasmata, and then diplotin is separation of the chromosomes. 89. Spindle fiber attached to kinetochore of homologous chromosome in metaphase 1 of meiosis, metaphase 2 of meiosis, both A and B, anaphase of mitosis. Here the right option is A, metaphase 1 of meiosis. 90th question. Identify stage, this one and this one. Option A is correct. This is anaphase 1 and then this one is anaphase 2. Meiosis 2. 91. Meiosis 2 initiated immediately after. Option A, telophase 1, B, prophase, C, cytokinesis 1, D. Chromosome have fully elongated. Here the right option is cytokinesis 1. After immediate when the cytoplasm is going to be divided off, then what happens? Meiosis 2 is initiated. 92. Movement of chromatid towards opposite pole is achieved by A. Shortening of microtubules attached to centromere, B. Shortening of microtubules attached to kinetochore, C. Elongated of microtubules attached to kinetochore, D. Elongating of microtubules attached to centromere. And then here the right option is option B. Shortening of microtubules attached to kinetochore. 93. Which of the following resembles with normal mitosis? Option A. Meiosis 1, B. Meiosis 2, C. Both, and then D. None of this. Here the right option is Meiosis 2. 91. In the beginning of Meiosis 2, a cell contains 4 chromatid. What number of chromatid is expected to be in each daughter cell at the end of telophase 2? It will be 4, 2, 8 and 16. It will be 2. 95. Find the mismatched column. Metaphase 2, prophase 2, telophase 2 and then anaphase 2. Which one is going to be mismatched? In the metaphase 2, chromosomes align at equator and then microtubules from the opposite pole of spindle get attached to the kinetochore of non-sister chromatids and nuclear membrane disappears. In prophase 2, nuclear membrane disappears. Is there one? Significance of meiosis. Conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organism is done by option A. Mitosis, option B. Meiosis only, C. Meiosis and mitosis, D. None of this. So here the right option is option B. Meiosis only. 97. Choose the correct statement about meiosis. A. Increase genetic variability of an individual of an organism. B. Decreases genetic variability of an organism from one generation to other. C. Reduction of chromosome by 1/4. And then D. Play an important role in evolution. It's very important that it play a very important role in the evolution. Which one? The meiosis. 98. The stage during which separation of the paired homologous chromosome begins is Pachytin, diplotin, diakinesis, zygotin. And the answer is diplotin. 99. Assertion and reasoning. Meiosis is also known as reduction division. Correct. Meiosis reduces the number of chromosome in daughter cells. So here the right option is A. 100. The centuryth question. And then last question of today's class, that is anaphase promoting complex APC is a protein degradation machinery necessary for proper mitosis of animal cell. If APC is defective in a human cell, which of the following is expected to occur? Means anaphase promoting complex will be there for the protein degradation machinery which is necessary for mitosis especially in the animal cell. If it is going to be defective, what happens? A. Chromosome will be fragmented. B. Chromosomes will not segregate. C. Recombination of chromosome arms will occur. And then D. Chromosome will not condense. So due to this defection, what happens? Chromosomes will not segregate itself, because in the anaphase segregation happens if that APC complex itself is defective, then what happens? Chromosome is not going to be segregated. Due to what happens? There will be many kind of DNA disorders will be there in that kind of humans. So this is the end of the session for today, that is the day 7, the chapter cell cycle and the cell division. For the next session we will discuss with a new topic. So for today, thank you and bye bye.